we move forward to our next speaker, Jennifer Gray from Georgia State University, from Neuroscience Institute as well. So we're looking forward to hear our next talk. Jennifer, floor is yours.
Thank you so much, and I'll go ahead and share my screen. All right, so thank you for the introduction. As she said, my name is Jennifer Gray, and I am part of the Forger Lab at Georgia State University's Neuroscience Institute. And today I'm going to be talking about the role of the perinatal vasopressin surge in neurodevelopment. So birth is one of the most dramatic events in a mammal's life, and all placental mammals undergo a shocking transition from an in utero to ex-uterine environment via vaginal delivery. And not surprisingly, birth is accompanied by dramatic changes in the newborn body. These include hormonal surges, activation of the immune system, mechanical stimulation via uterine contractions and going through the birth canal, exposure to microbes in the vaginal area, and independent breathing, thermoregulation, and water balance. So the fetus has presumably evolved to develop a way to cope with all these very rapid and shocking changes that are happening at the time of birth. And one such unique but poorly understood event in the newborn is a striking surge in peripheral vasopressin at the time of birth. And actually, previous research has shown that humans experience a near 100-fold increase in vasopressin following vaginal birth, with levels that are never again seen throughout the lifespan, even in the face of challenges later in development. However, the neural underpinnings of this vasopressin surge in the neonatal brain remain largely unknown to our knowledge. So we first decided to assess widespread neuronal activation following a vaginal birth. And we found a particularly striking effect in certain hypothalamic regions, including here, if you can see my cursor, the paraventricular nucleus of the hypothalamus, the supraoptic nucleus, and the suprachiasmatic nucleus, the SCN, which play a principal role in homeostatic and hormonal functioning, as many of you know. And we found that neural activation, which we measured here using cFos as a marker for neural activation, peaks 3 hours following a vaginal delivery. And we are particularly interested in the PVN, as this houses a large homogeneous population of neurons which are involved in hormonal regulation. So we then wanted to see what specific neuronal populations were activated in the PVN following a vaginal delivery. So we stained for 3 main cell types, as you can see here on the right. We stained for vasopressinergic neurons, which we labeled in green, oxytocin neurons, which are here in blue, and corticotropin-releasing hormone neurons labeled in red. And we looked at 3 different time points, E18.5, which is a few hours before birth. As many of you know, the mouse gestation length is 19.3 days. 3 hours following birth, and birth is marked by the red dotted line there, and at P1. So we found that activation of vasopressinergic neurons specifically more than tripled 3 hours following a vaginal delivery. And this suggests to us that birth itself may be triggering activation of these vasopressinergic neurons in the hypothalamus. So we then wanted to see how vasopressin was being expressed centrally around the time of birth. So we analyzed PVN punches from embryonic days 16.5, 18.5, as I mentioned previously, and 19, just a few hours before birth, as well as 3 hours following vaginal delivery, and then at postnatal day 23 during periadolescence. And this was done using qPCR. So we find, incredibly, that vasopressin mRNA expression is significantly increased prenatally and is elevated in the early hours after birth here at 3 hours. However, expression levels drop drastically by P23. So this indicates that the fetus may be upregulating vasopressin expression in preparation for these challenges it faces at birth. And as I mentioned previously, birth is accompanied by these incredibly dramatic stimuli. So this surge in vasopressin may have a functional role in both peripheral and central nervous system function. Now, one unique aspect of human birth is our ability to manipulate birth mode by performing cesarean sections, as you can see on the right. However, obviously, the neonate's experience of birth itself is significantly altered during a C-section, and many of these stimuli that accompany a vaginal birth, like going through the birth canal, exposure to these microbes, and even these surges in hormones, are significantly altered or absent in C-section births. And one of these events that is significantly altered by birth mode is this peripheral surge in vasopressin. So actually, previous research has shown that plasma vasopressin levels are significantly lower in C-sectioned newborns. However, C-sections are becoming increasingly prevalent worldwide, and in the US alone, 32% of all births are via C-section. So taking all this into consideration, we hypothesize that the perinatal vasopressin surge may actually aid in this transition to an exuterine environment by impacting both peripheral and central functions. And we think this primarily may be acting in two ways. One, by increasing water retention and overall hydration in the neonate, and by influencing neurodevelopmental patterns at birth. Finally, these functions may be significantly altered by birth mode. So we first wanted to establish if peripheral vasopressin is actually altered in the neonatal mouse and how this looks at the 3-hour time point following vaginal delivery, where I mentioned previously vasopressin neurons were most active. So using copeptin as a one-to-one marker for vasopressin, just as it's a more stable molecule in plasma, we find that copeptin was significantly higher in vaginally born animals compared to cesarean-sectioned mice. So it is known that vasopressin functions largely to regulate water balance and plasma osmolality by binding to V2 receptors in the kidneys and just overall impacting renal water retention. And just as a refresher, plasma osmolality, as it decreases, water retention and blood volume increases, and vice versa. So to assess the impact of this vasopressin disparity between the two birth modes on overall hydration in the neonate, we measured plasma osmolality of vaginally born versus C-sectioned mice. And as you can see, overall, regardless of birth mode, both vaginally born and cesarean-sectioned mice exhibited lower plasma osmolality compared to what is considered the normal range. However, vaginally born mice, which you can see on the left, showed significantly decreased plasma osmolality in comparison to cesarean-sectioned mice. So this indicates that something at birth, particularly related to birth mode, is impacting water retention and hydration in the newborn. Finally, it has been established by our lab that birth is accompanied by a striking change in region-specific cell death patterns. Specifically, we have shown that in the PVN here, there's a significant drop in cell death at 3 hours following a vaginal birth. It starts dropping at 1 hour and it's most significant in 3 hours. However, this effect is transient, and at 6 hours, cell death rises. Here we used activated caspase 3 as a marker for neuronal cell death as well. We also found that birth mode impacts cell death in the neonatal brain. So specifically in the PVN, we collected vaginally born and cesarean-sectioned mice 3 hours following delivery. And in the graph on the left, you can see that at the 3-hour time point, vaginally born mice exhibit that drop in cell death. However, that drop in cell death is largely absent in C-sectioned mice, establishing that some aspect of vaginal birth may actually protect against cell death in this window around 1 to 3 hours following birth, and cesarean-sectioned mice are lacking that stimulus. So to summarize, following a vaginal birth, mice exhibit higher plasma vasopressin and a decrease in cell death in the PVN. And conversely, following a C-sectioned birth, there is lower vasopressin in the periphery and an increase in cell death in the PVN. So this begs the question, is vasopressin itself neuroprotective? So to test this, we performed C-section deliveries, and at the time of birth, we injected into the ventricle either vehicle or vasopressin, and then 3 hours following injection collected the neonatal brains. And incredibly, we find that vasopressin injections do indeed reduce cell death in specific hypothalamic areas. So looking at the PVN, we see that C-sectioned mice that received vasopressin injections exhibit significantly reduced cell death density in the PVN compared to C-sectioned mice that received vehicle. And similarly, in the anterior hypothalamic area, we see that C-sectioned mice that receive vasopressin injections also exhibit that reduction in cell death that we are not seeing in the vehicle mice. So, conclusions. Neonates do experience a surge in vasopressin around the time of birth. Additionally, vasopressin may be neuroprotective and defend against dehydration in the periphery. And finally, altering birth mode does alter the perinatal vasopressin surge as well as neuronal cell death. And this might actually have possible long-term effects, not only on neurodevelopment, but possibly on behavior later in life. So I just want to thank a few people, one of them being Laura Cortez, who just went before me. Of course, Nancy Forger, who is my incredible mentor at GSU, Dr. Alexandra Castillo-Ruiz, another incredible mentor and role model in our lab, and Dr. Ureli Hoffitz, who spearheaded the majority of these projects, as well as Taylor Height, Megan Hall, and Emma Ramlall. And of course, the GSU NI core facility, who helped with so many of these experiments as well. Thank you, and I'll take questions.
Thank you very much. This is exciting, all of this, the studies that you do, guys, in the lab. It's amazing. I'm just mind-blowing. Do you have any questions? Participants? This is, it's mind-blowing. Definitely, so many questions should appear. So for me, the first question would be, are there any long-lasting effects?
So actually, we have preliminary evidence that suggests yes. We are looking at vasopressinergic neurons at, I believe, postnatal day 84, so into adulthood. And our preliminary evidence does suggest that there is a difference between the two birth modes.
Okay.
So that's pretty exciting, yeah.
And do you think there's also difference, for example, for primates?
Yeah, that's a great question. So this vasopressin surge and disparity between the birth modes has been observed in, I believe, sheep, obviously in humans. And yeah, I mean, it seems to be a conserved mechanism. So I believe that in other species, this could be present.
Okay. Do you think it's like possible to do something as a treatment or going for some sort of compensation for loss of the vasopressin?
Yeah. Our lab has not looked into that yet. We're just trying to establish, the mechanisms at which it's working. But definitely, I mean, this could have very, incredible clinical applications as well. And, it's surprising there's a lot of research done on the disparities between C-section and vaginally born neonates, but a lot of the research is done in the mom, and the neonatal brain, as I said before, has been largely neglected. So our lab is trying to, figure out what about a vaginal birth, whether that's, microbial exposure, vasopressin surges, immune activation, what about vaginal birth may be protective.
Okay, great, thanks. We have a question here from Kathy. Great talk. Did you observe any liter-to-liter difference in vasopressin, the surge? Have you considered any emergency cesarean condition? Example, gamine, dystoxia. Yeah, sorry.
Yeah. No worries. Yeah, that's a great question. So obviously the conditions under which a C-section is performed can widely vary depending upon whether it's an elective C-section versus an emergency C-section and a traumatic birth. And I'm sure that during a traumatic birth, you're experiencing a lot of peripheral and central inflammation, and that could definitely impact cell death and neurodevelopment. We haven't looked into that yet, but we definitely are looking into the immune activation side of birth.
Okay. Well, thank you. So now we go to the next.